As always, please try and support our National Neurotrauma Society and join or renew your membership. That's greatly appreciated. So we're delighted to have Michelle here today, and also it's very special that her fellow, Lauren Ryder from Utah, is also going to be introducing and moderating this session. So we're really looking forward to this. And thanks, Lauren, for taking this on, and we look forward to your talk, Michelle. Thank you.
Alrighty, thank you for having me. So today we'll be hearing from Dr. Michelle Schober. She is an associate professor of pediatric critical care medicine at the University of Utah and an attending physician in the pediatric intensive care unit at Primary Children's Hospital. We've had the pleasure of working together closely in the clinical and laboratory settings as our passions for neurocritical care align. And today she will be giving a talk on her research regarding DHA and pediatric TBI.
Thank you, Laura. And thank you very much to Adam and Josh and David for organizing this wonderful opportunity. I'm very appreciative. And with that, I'd like to introduce you to my topic. I'm going to talk to you about pediatric traumatic brain injury and the potential role of docosahexaenoic acid from here on forward, called DHA, a potential neuroprotectant. I have no disclosures. And I did want to start with a brief overview. The audience is quite familiar with the impact of traumatic brain injury in general, but in children, this is particularly significant, certainly to me and to society, because of the number of deaths. But not only that, but severe pediatric traumatic brain injury, the kind that results in the patients that I see in my intensive care unit, as you can see shown in the picture, it's estimated about 2,000 patients per year, and sadly, half of these severe TBIs, as many as 50%, are discharged alive with major disability, and that actually is estimated to result in 1.3 million life years, again, because of the life expectancy of our children. So unlike many other diseases that I see in the intensive care unit, where I provide very similar modes of support, the ventilator and whatnot, I have no tool, well, no one has tools to actually treat the pathophysiology. So that drives me to conduct bench and translational research to try to find something other than supportive care to help improve this disability. Age and injury is important. It actually, when you compare across the age span, there's certainly looking at the pediatric range, the younger age is worse, even accounting for factors that are social or cultural. And it may well be that there's something about interrupting that normal development. In animal studies, it's actually supported as well. It's very difficult to model exactly, but trying to compare injury severity across ages, it also appears that there's a vulnerable period of brain development that is an important factor. What are some things we can actually modify, however? So of interest to me is that the developing brain is very vulnerable to oxidative stress and inflammation. The reasons for that is that on the one hand, there's developmental delays in our ability to protect the brain. That refers to having a slower development of antioxidant enzyme systems and also the small molecule antioxidants, and that's been well reviewed in the literature. And the other factor is not unlike what Dr. Bickford was describing in the last seminar. At the extremes of age, sometimes things meet, and so there's a tendency towards a more vigorous inflammatory response at the younger age of the spectrum. Animal studies support this. For example, there's a CCI model where you have 21-day-old mice. They're just about ready to wean from the mom. And compared to the adults, they had about 2 weeks of what seemed to be monocytes, or infiltrating macrophages or activated microglia, whereas adults had about 3 days worth of it. And certainly studies where you have comparing weight-adjusted dosing of, say, LPS or inflammatory agents, having a more vigorous response in the younger brain. And even basal levels of inflammatory cytokines certainly seem to be higher. So there's reasons to think that we could think about microglia. And again, at the ends of the spectrum, and also in TBI, microglia have recently been found to be important in terms of trying to improve outcomes. So it's well documented that the production of free radicals, including superoxide anion via the NOX2 enzyme, which can result in hydrogen peroxide and further in the presence of transition metals, which is particularly important in the absence of the methylmethionine that we talked about with the developmental delay. You have the very toxic hydroxyl radical. Also, the microglia can produce nitric oxide via NOS2 synthase, which in the presence of superoxide become peroxynitrites, again, very toxic to the brain. So we certainly will use the M1 terminology, knowing that this is not by any means simple in terms of inflammation, but quote unquote M1 markers found on microglia, they're associated, again, not specific for production of free radicals. And the other source of pro-inflammatory effect, of course, are cytokines, including TNF-alpha and IL-1 beta. But then conversely, microglia can also promote repair. So microglia can promote the birth of new cells, axon repair, and remyelination. And the action of phagocytosis is actually necessary for oligodendrocyte recruitment, and secretion of neurotrophic factors such as TGF-beta and IGF-1 is also important. So once again, without oversimplifying, microglia that express M2 quote-unquote markers are associated, albeit not specific, for phagocytosis and repair functions. So it seems to make sense to try to see if we can support sort of the transition towards the M2 phenotype in an attempt to promote repair. So I was drawn early to DHA, primarily because I knew of its role in the cardiovascular system and also in macrophages and fat in terms of promoting an M2 phenotype. So as I read into it, I realized that DHA can modulate neuroinflammation in many degenerative diseases such as Parkinson's. And in vitro, it's certainly been shown that it can modulate microglia and macrophages towards the M2 phenotype. So DHA is a component of fish oil. You can see the molecular structure up in the upper right-hand corner. And it has indeed been tried in adult TBI models, and it seemed to improve standard neurologic outcomes. And there's many more molecular studies since the very first ones, but ultimately it's currently in phase 2 in adults to be used after mild traumatic brain injury. And the outcomes at this point are cognitive and serum biomarkers, but they are randomized controlled trials. So in the adult world, this has made some advance. So of course I asked myself, well, what about traumatic brain injury? And certainly at that time, there was nothing at all. And even now, there's very scarce literature on DHA and TBI. So back then I had a pretty old fashioned model for CCI, controlled cortical impact. And you can see in the diagram on the right that the impact lands on the cortex and produces some hippocampal damage. And this is a model many are familiar with, and it's well described in the 17-day-old rat pup. I chose that age. A, it's an established model, and B, it models the age of worse outcomes in pediatrics, which is the infant to toddler age. It's particularly at this time synaptic pruning, synaptogenesis, myelination is ongoing, neurogenesis is pretty much done. So at that time, I tried a 0.1% DHA diet. I came upon that because one of my colleagues in the lab was studying DHA as a treatment for, well, maybe not treatment, but anyway, as a PPAR gamma agonist in intrauterine growth retardation. And she tried a number of different diets, and that was the one that produced the PPAR gamma effect, and more did not have any more effect. So I had the very same diet without DHA, which I'll call regular diet, and then I had my sham controls. So I was somewhat surprised actually that it seemed to actually help. The rats were injured at 17 days of life, as I said, and they were exposed to Morris water maze about 45 days later after injury, as you can see on the horizontal bar. And they had improved performance. The black bars are the regular CCI, and orange is the DHA treated, and the sham is in white. And similarly, the lesion volume at 3 days after, shown in the A labeled horizontal bars, DHA had less of a lesion than did the regular treated rats. And similarly at day 50, so after the Morris water maze, they also had lesion sparing. So I then, thinking about the oxidative stress, compared the production of nitrites and nitrates in tissue, and here shown in black are the regular CCI rats, day 1 cortex, day 2 cortex, and DHA indeed seemed to be protective. So I proceeded to conduct MRIs on them and found that overall at 12 days after injury, the regular CCI rats had generally, more edema as shown by T2 imaging than the DHA rats. And then I looked at radial diffusivity as a marker of axonal injury. Just to refresh everyone, the radial diffusivity would mean that there's more water movement perpendicular to the axon, suggesting breakage of these white matter injuries. And then I did find a difference between DHA and regular, again, limited to the body of the corpus callosum. Nevertheless, finding some improved white matter injury. So along the lines of inflammation, I had found the oxidative stress difference, but I wanted to do a temporal screen for inflammation. And here, thanks to my collaborator, Dr. Pauly at the University of Kentucky, we did some autoradiography with TSPO binding, where more TSPO binding suggests more neuroinflammation, which initially was thought to be specific to microglia, but it's now clear that it could be done by astrocytes as well. But it's a well accepted measure for neuroinflammation. And in green, you can see the areas with more inflammation. So as you would expect, the sham animals don't have any. This is a baseline TSPO binding that is disregarded. And the CCI on the side of the injury all have some inflammation, particularly day 3 and 7, and waning after that. But DHA did not make any difference. So I was a bit perplexed by that after having seen the oxidative stress protection. So I proceeded to look at tissue. And so here I looked at the hippocampus and looked at tissue. And shown in black are the regular CCI rats, and DHA treated are in shaded gray. And there I looked at some, again, quote unquote, M1, M2 markers in tissue. And on the y-axis is simply mRNA expressed as 2 to the delta CT method. And there, remarkably, this is done at day 2, so the first half of the graph, just to guide you a little bit. I have day 2 and day 3, one after the other, so TNF alpha and TNF alpha, but the day 3. Generally, the effects were more pronounced at day 2. The asterisks show the difference where DHA is having a decrease. So in the case of TNF alpha, DHA was protective. It did not have any change in TNF alpha by day 3. This is chemokine receptor. So this is sort of a signaling measure of signaling for macrophages to infiltrate. And here DHA again was protective at day 2, but not at day 3. Interleukin 18 accessory protein and the interleukin 18 again at day 2. And I also looked at IL-1 beta, and here by day 2 and day 3, DHA also appeared to decrease relative to regular treated CCI rats. Some of these markers, I found such very low mRNA levels. It was hard, the variability was very high, but did not seem to be much of an effect in terms of interferon gamma, INOS, or interleukin 6. And then hippocampus at the ligand for so CCL, chemokine, CC chemokine, this is the same thing as MCP1. Basically here, similarly at day 2, DHA decreased the expression of that ligand, but not at day 3. So I was encouraged, but realizing that my model was not exactly, and I forgot to explain a little bit. I gave the DHA to the mom the day before injury. And the reason I did that is because the pups were so little that they would not, I was worried they would aspirate if I gavaged them. So knowing that at least in humans, it's about 12 hours for the composition of maternal milk to change, I fed the moms the DHA diet the day before. But again, that's not very translatable now, so I did not have a sham DHA group. So I modified my model, moved with a impact still at day 17, same site. The difference here is that I injected them with DHA diluted in intralipid, and the regular treated rats received intralipid and vehicle I went that route. I tried a couple of different things, but ultimately sort of estimating what they would normally ingest and then translating that into how much DHA that would be. I came up with some doses and checked DHA content in the blood and the brain, and ended up essentially giving the rats 7 milligrams of DHA injected intraperitoneally on the day of surgery, so 30 minutes later. And then I would change the diet for the moms, and then no further parenteral administration. So with that, I repeated my nitrates and nitrites and showed the same effect. This time I combined hippocampus and cortex, knowing that my mRNA results, which I also didn't quite explain, but the cortex was so injured that the mRNA markers weren't changed by anything. And so it seemed to make sense to take the ipsilateral hippocampus and cortex together to try to get signal that wasn't quite so different and still reflected injury. So with that, I then moved on to design a study over time. And here, I was more interested in separating the cells out rather than the anatomic location. So what I did is I decided to look at flow cytometry. So taking the cortex and hippocampus, making a single cell suspension, and using flow cytometry to separate the 3 populations defined as negative for 11B, which would basically encompass neurons, astrocytes, endothelial cells, certainly quite a mix. However, the 11B positive population primarily would be composed of macrophages, microglia, some neutrophils based on mouse literature. But as many of you know, the paucity of rat antibodies limited me somewhat. And so I ended up selecting 2 populations, CD45 high to be enriched in infiltrating macrophages, and CD45 low to be enriched in microglia. And I defined my high and low using blood, where CD45 high was the neutrophil signal. And so then I also looked at histology for markers for M1, M2 markers in microglia and astrocytes. And I selected 3, 7, and 30 based on the TSPO data. So here, in collaboration with Dr. Ginsell, the University of Kentucky, we found some interesting results. What you can see on the left-hand side is the intensity of staining for IBA1 in the cortex. And black bars show regular CCI, white bars show DHA CCI. And clearly the 2 sham groups are pretty similar. And this is shown also in the picture. So here DHA decreased microglial activation as shown by IBA1 histology. And here at day 7, there was an injury effect. Clearly both groups had higher activation, but DHA did not have an effect. Then I tried to look at the, instead of the cortex here, now it's the hippocampus, and their injury effect at day 3, but not a DHA effect, which is logical in light of the expected intensity of injury being higher at the cortex than the hippocampus. And I look at CD68 as an M1 marker together with Dr. Ginsell, and here there was a more dramatic difference seen, again, more strongly at day 3, but with a DHA diminution of response, and day 7, continued ongoing injury effect, but no effect on DHA. So as far as this image, I just wanted to give you a sense of how the gating was done. And so I don't have the flow pictures for the blood here, but what is drawn for the gates are based on blood. So these 2 graphs on the left are sham animals, sham regular at 3 days after injury, and sham DHA at 3 days after injury. And the gate for CD45 high shows very few cells. Whereas the 2 injured groups, here you have, in the 3rd panel, you have regular treated CCI at day 3, and the last panel is DHA treated CCI. And here in the CD45 high range, suggesting infiltrating microglia, clearly an increased number. So that was how the things looked. And then what I did after the cells were sorted out, it took a long time to get it to, have cells that were alive enough to produce RNA. So I ended up going the nanostring route so that I could make the most of the RNA that I got. And I selected markers purely on literature and my best guess. And I picked some transcription factors that I thought would be of interest. So related to the interferon family, IR5, 5, and 8 are important for macrophage and for microglia, respectively. And PPAR gamma 1 and 2, because I continued to think that I would find a DHA effect. I did not find any real difference here. Let me just walk you through. The black is the regular CCI and the white is the DHA CCI. And there's no shams in this picture because the other difficulty that I had is that the injured animals clearly had enough cells in the high region, as you can see from the previous pictures that I showed you, but the shams just did not have enough. So for studies other than the negative cells, I only compared the injured groups to each other. So here, no statistically significant difference really at day 3, but at day 7, there's a big injury effect, a DHA effect for both IRF5 and 8, where regular rats have higher levels. These are normalized to log RNA counts. And also to explain a bit more about nanostring, it was normalized to housekeeping genes as well as to the positive controls. So in the negative cells, as I said earlier, I had more to work with. I had sham RNA as well. And what you can see here, again, on the vertical is the log RNA, and the horizontal are a number of markers associated with M1 and M2 types. And here, sham is in gray. So many of these are abnormal due to injury. Those are all shown by the asterisks. The pound signs refer to the ones where DHA lowered the expression. This was the negative group. I put that up for illustration, but again, because I had the sham animals, but I was very drawn to this set of data. So the low, which would be enriched in microglia at day 7, here there's no sham group, it's DHA and CCI only, but all these asterisks refer to differences in DHA versus regular. So again, there seemed to be something about day 7 at which time DHA had more of an effect. And generally speaking, what you see here on this side of the panel are M1 associated markers, and these are M2. And similar to what others have found, it actually seems to be that when M1 goes up, so does M2. So I found, for example, just to give you an illustration, here's interleukin 10, where regular had more than DHA. And then you look at markers like interleukin 1 beta, and regular has more than DHA. So basically, regular had higher of both in general, M1 and M2 than DHA did. But the biggest, most significant set, I suppose, was in the microglia population at day 7. So in summary, I found that DHA improved lesion volume, white matter indices of injury, cognitive function. These are all male rat pups, 17 days old after control cortical impact, and it didn't seem to matter whether the DHA was given before or after injury. And found that DHA appeared to decrease microglial activation early after CCI, and at day 7 after CCI, using cell-specific RNA, DHA decreased microglial M1 mRNA and the transcription factors 5 and 8. So with that information, I decided, well, it is now time to look at females, and it is time to look at protein markers. I figured the only way to bypass the sham problem with RNA plus protein should be more important. So I have been doing protein studies using the same approach of flow cytometry, same 11B separation. And so far, I'm happy to report that the protein over time, this is IL-10 and this is CD68, follows a very similar pattern. This is just sham and regular, but injury effect is very much tracking along the line that I saw with mRNA. And here I looked at some DHA effects, and I wanted to see, again, would it track along? And so far, IL-10 does seem to be blunted by DHA. DHA here doesn't show up very well. It's actually the 3rd column. And the black is regular CCI. The 2 sham bars are shown. And clearly an injury effect increasing interleukin 10, an injury effect increasing CD68, and DHA, especially in macrophages, decreased by DHA. So far so good. I'm still working on collecting data. And then female and males, I've also started with that. And here, very preliminary, but interestingly, it seems that the CD68 expressed as percent of cell type. Here you have regular CCI males, and then the bar next to it is sham regular males. And there's no DHA on this graph, it's just the regular females shown on the third bar, and the fourth bar is sham regular. So basically the females seem to show lower percent positive CD68 and also CD206. But that's not universally true. For other markers like TNF-alpha, injury effect predominated. Here, the black and the gray have similar levels. And TGF-beta was opposite, where it didn't really seem to matter so much whether you had injury as the sex mattered. So for whatever reason, the TGF-beta seemed to be lower in females than in males, regardless of injury. So what I haven't yet started, but I'm looking forward to doing, is looking at functional effects on microglia. And it's hard because in vitro doesn't recapitulate in vivo, but then once, you know, if you really want to see what they do, it's kind of hard to do that in an animal. So thanks to Dr. Lone and Duran, who actually have been very kind and spent a great deal of time with me on the phone. And I've been reading a great deal about what Ritzel's been doing. In fact, his most recent publication was of great interest to me as well. So I will soon be doing some pilots to see how reactive oxygen species are affected or not by sex, by CCI, and by DHA. And similarly, phagocytic capacity, also with their guidance. And I have a plan to try to quantitate the effects. So this is the general study design. The idea here is that at 30 minutes, they get injected just like I've done with the males, but now I have both sexes. And I'm picking 1, 3, and 7, and essentially looking at protein FC markers, the activity that I mentioned, and continuing the DHA diet just like I've always done, looking to see function. And this time I'm adding, well, not adding, but combining measures I've used before: novel object, Morris-Water maze, MRI, histology, and males and females. So a lot of this is to try to see how males and females compare previous to my other findings. But I also, as a physician, you know, would like to know, does it have to be 30 minutes? Can it be longer? So I have a separate arm where I'm going to inject some at 3 hours. And the other thing that matters to me is, you know, is it possible that DHA just needs to modulate inflammatory response in the first 7 days? Since based on TSPO, inflammation seems to wane after that. And I know that DHA has other effects, including a building block, so to speak. It's part of the brain membranes. And so is it possible that just 7 days is enough to achieve good outcomes based on function and MRI and histology, or do I need to continue 60 days? So this is essentially the same design. The only difference is that because I did not include mRNA in my previous study, I'm gonna do some of these as well, just to kind of have a better sense of the whole picture. And I have a hypothesis finding sort of arm where trying to put all this together of how I think DHA works. To me, a unifying hypothesis, at least in my model, is that DHA gets incorporated into membranes. I have other studies showing that DHA does get lost after TBI, and the ATP deficit is such that it's hard for the brain to basically re-esterify DHA right after injury. So when the rats are fed the DHA, they're able to replete their DHA content sooner. So the idea here is that DHA, by incorporating itself into membranes of macrophages and or microglia, that they'll affect the lipid raft composition and potentially the signaling of toll-like receptor 4 and others, and that ultimately that might decrease damage-associated molecular pattern signaling, which is known to be important in TBI, at day 7. It's a way for me to sort of make sense of my data where I see an effect later after injury. So here's a little diagram. TBI happens, the damage-associated molecular patterns, including high mobility group proteins attached to toll-like receptors, 4 in particular, and interferon gamma receptor. lipid rafts are regions that are known to be important for aggregation of proteins and to actually signal. So the idea here is that toll-like receptor 4 signaling, we know it promotes translocation of the IRFs 1, 5, and 8. And if there's more translocation of IRF8 to the nucleus, there should be more expression of inflammatory factors. And by the interferon gamma receptor pathway, JAK-STAT activation signaling will increase the production of IRF8 mRNA, which would go along, at least if this is actually what DHA is doing, blocking both of these, it would fit with my decreased IRF8 mRNA. The ultimate result of both of these steps is more inflammatory cytokines, TNF-alpha, interleukin-1 beta, and 6, just to name a few. So this is the gray signaled areas is what I'm focusing on. But what I intend to show with this diagram is that DHA is pleiotropic. And certainly it's well known that DHA acutely it would be free probably in terms of not being esterified, and that any presence of DHA can produce derivatives, some of which have been found to mediate inflammation directly. And then chronically, there's studies in different models showing effects on membrane fluidity, synaptic vesicle translator release. But for me, for my interest in children, DHA uptake is very high in the first 3 years of life. And so if a child's on a Western diet and they have DHA losses from TBI, it may well be that, this is particularly important to treat children for longer. But for this purposes of my study right now, I am looking at the subacute pathway, as I just explained. So this is just to give an idea of how I can test this hypothesis. I would have to basically treat the models the same way, but this time extract brain to make the lipid raft fractions, and then look at Western blots for my, my markers of interest. I'm also going to do histology for colocalization of phlotolin and toll-like receptor 4 as a marker for disruption of this of toll-like receptor at least expression. And also whether or not there's translocation to the nucleus of phosphostat, looking at colocalization with DAP. Caspase activity is an indirect marker of DAMP signaling. So I have some preliminary data, at least I can isolate the rafts, and I was able to measure toll-like receptors. So this is all very preliminary, but I'm looking forward to exploring this hypothesis. And then just finally, just wanted to let you know that Dr. Floyd at the University of Utah and I have worked together. We're appreciative of some seed funding from the University of Utah. And we set up a piglet model of pediatric traumatic brain injury. Lauren actually has been helping me with this. And the idea here is that as a physician, clearly I need to, well, I want to move towards my patients. And rats would not allow me to get the data that I need. So the actual hypothesis in terms of DHA effect is very similar. The difference is that I'm looking at biomarker changes, and I'm looking at cerebral spinal fluid and blood and urine, just like I would in a human patient. And the other thing that I can do is that I can look at oxidative stress biomarkers, because I do worry that maybe DHA will hurt in terms of worsening oxidative stress. So I can actually measure free radical injury in the brain of the piglets, but also look at markers of oxidative stress to see if that's a way that I could design a future clinical study. So here is the basic diagram, the injury, the animals, and how I would look at oxidative stress in terms of fresh brain, but then here look at biomarkers and of course traditional histology. So that's the little reservoir to remove CSF serially, which is sort of similar to what we do in kids. And I have some preliminary data, at least I can induce injury, or we can. So Flora Jade showed some neuronal death. But this is my ultimate goal. Again, I would like to approach the bedside with more than these wonderful tools, not to downplay them, but something that I could provide that would actually help kids have better outcome. So with that, I'd like to thank my collaborators, Dr. Polly, Dr. Gonzell, the University of Kentucky, Dr. Lone, who's been so helpful with his advice, and funding from the NIH, the Division of Critical Care, and the University of Utah for their seed funding. Thank you for your attention, and I'm ready to hear if you have any questions. Thank you.
All right, awesome. Thank you, Michelle. That was excellent. We're going to use the Q&A feature on Zoom to answer any questions. So if you guys wouldn't mind typing your questions into there, and then Michelle will answer them for us. I guess just to kind of start everyone off with the questions, this is more of a broad question for Michelle, but it seems as though this kind of one research question that you started with has really kind of developed into this almost like career's worth of research. And I guess as a junior researcher, what kind of advice would you give to those of us who are just kind of starting in our careers as far as like how to find your research niche or develop a good question that can really turn into something as great as your work?
Well, thank you, Lauren. That's very kind. I think actually, to quote my division chief, it has to actually matter to you. He has a different word for how something matters to you, but it should really matter. And I guess to me, it always seems like you break a leg or you have, something that the body is going to be okay. The brain has always frustrated me as not being so resilient. And then I guess in terms of your question about how to find a niche, that's a really good question. It's hard because sometimes the environment around you will take you somewhere. I just don't necessarily think that there's the one particular spot to be in, as long as it's related to something that actually matters and you're taking advantage of what's around you. And as long as you look at the data with open eyes and not trying to make it be what it isn't, I think people will end up doing good science, and finding a spot that is relevant is so much easier when you care. I don't know if that's helpful, but that's what I've done, and that's been good for me.
No, thank you. That's great. We do have a question. I'm going to go to the answer live function, but what are your thoughts on another omega-3 fatty acid EPA in comparison to DHA?
That's great. That has definitely been important to me. I've been quite careful actually not to mix them because they are different. Let's see, at least in terms of the limited data where you're actually trying to separate it, it appears that sort of that M1, M2 effect, which is what at least I've been seeking, DHA appears to be the primary driver for that. EPA definitely has mediators that are neuroprotective, and I have no reason to say people shouldn't potentially consider both. I was just trying to kind of keep things a little separate because they do have different effects in terms of phenotype and microglia in vitro.
Okay. All right. So we have another question from James McAllister. Would DHA be as effective with repetitive mild TBI?
That's, thank you. I actually think that the studies in adults, since they're being done with mild TBI, there's a certain appeal to giving it with mild TBI in the sense that the worry that I shared with the audience about could it be that I'm going to worsen oxidative stress by giving it after such severe injury would probably not be relevant in mild TBI. I would think that at least the mild TBI models do show benefit. And so if anything, I would think repetitive should, since usually the outcomes are worse with repetitive than single, it should help. But I'm not aware of specific, in fact, I don't know of any studies in pediatrics other than mine and some limited ones. And I don't think any of those are with repetitive mild TBI.
Do we have any other questions from the audience?
Michelle, I'll hop in here and ask a quick question.
Yeah.
Have you looked into any of the resolvins as a potential mechanism for why you're seeing the suppression of the inflammatory response after DHA?
Thank you. No, that's something that I just haven't had the bandwidth to do, but it's certainly been, I know that there's a great many publications on that. No, I have not, and that's certainly an avenue to pursue.
Anyone else with any last minute burning questions? Ooh, there is one. There is a question regarding, is there any evidence that TBI outcomes are better for people who live in countries that consume a lot more fish than we do here in the US?
That's an interesting thought. I actually don't even know how one could begin to do that, because we haven't done, I'm not aware of any studies giving DHA to people other than what I mentioned that's actually right now still enrolling. So you'd have to have a study where you randomize it, and yeah, no, I'm not aware of any of that. And TBI outcomes are so multifactorial that I could categorize that by fish. I did design a study that didn't get, the high school athletes didn't want to get blood taken, but the study design included a dietary questionnaire to figure what their baseline DHA intake would be. And we were going to give them DHA and have them go through a training season and all kinds of things. So I was trying to answer that question, but I haven't been able to yet.
Well, I'll hop in with another question while people are thinking. How do you think the balance of omega like 6 to omega-3 fatty acids in a diet, or just, you know, you mentioned American diet and more high in saturated fats versus omega-3 fatty acids. How do you think that might affect TBI outcomes? Do you think omega-6 would be worse than omega-3 if you have a balance that's not correct?
Yeah, well, that's another good thought. I guess I would say that not all omega-3s are created equal. And so similar to how I said about the DHA and the EPA, but the bulk of, I guess, the 6 in the diet would be arachidonic acid. So certainly people who've done research in the so-called cafeteria diet or Western diet show far worse cardiovascular outcomes, and that's pretty well shown. So I could only surmise that in the same way that the brain studies seem to parallel the cardiovascular in terms of inflammation, that there should be more inflammation. But I haven't actually actively looked to see if there are studies on Western diets and TBI, to be honest. Most of them have focused on the cardiovascular system.
Michelle, David here. Just a quick question. Sorry, I may have missed this because I lost my internet connection, but have you looked at IL-4 signaling or STAT6 dependent signaling in DHA? Is there any evidence to suggest that, you know, that DHA is driving this M2 transition through that signal transduction pathway?
So through STAT6, that's certainly been something in the M1, M2 phenotype. I could not find anything specific to STAT6. And as far as DHA affecting interleukin-4 expression, I do think that it has been shown in other systems like fat and cardiovascular system. Definitely not something that I'm aware of in TBI. But the reason I chose to focus more on the IRFs is I was looking for microglia macrophage M1 modulation, but there may be literature on STAT6 and other cell types.
Thanks.
Any last minute burning questions for Dr. Schober? Okay.
All right. Well, thank you again. It was a pleasure to be able to present my data to you. Thank you.
Thanks, Michelle.
All right.